Okay, apologies for the delay. It's evidently an issue of safe sharing knowledge. We were somehow prohibited by internal firewall problems, and that was causing the delay. My name is Thomas Steger-Hartmann. I'm head of investigation and toxicology at Bayer, and I want to talk about our experiences with regard to safe sharing of safety data in two IMI projects, namely eTox, eTransafe. Next slide, please. I would like to talk to you about the need for data sharing. So I assume, I do not know what your audience actually is, or what your professional background is, but I assume if some of your background is toxicology, you already know why there is a need for data sharing. But I would like to go back a little bit in time in the first couple of minutes of my talk to illustrate where we come from and what we have achieved over the last couple of years. And then talk about the prerequisites for safe sharing, both from a legal and an IT perspective. Look at what can be done with the data we share and what are the learnings from IMI, eTox, and eTransafe. IMI stands for Innovative Medicine Initiative. Now, next slide, please. One of the most known the reasons for data sharing lies in the field of mutagenicity, particularly in the pharmaceutical industry. Mutagenicity is a chemical reaction with heavy consequences for drug development. So if you have an Ames positive, normally you stop the development. I'm just showing here one of the typical reactions where we have to cope with, and the slide is animated. So if you click once more, if you collect all this data which is in the literature, this sets the stage for developing expert system. Collecting all this information from the literature led to the seminal publication in 1988 by Ashby and Tennant, which shows all the critical elements, critical moiety, which could lead to an Ames positive compound. And this is the onset of in silico systems. And if you click on the next slide, slide number 4, in the meantime, we are really good at modeling mutagenicity. So it works, but the extraction of the data from literature was cumbersome. There was substantial effort in standardization and curation, even though we are talking about a relatively simple in vitro screen. And then another issue was that the focus in this publication was on Ames positive compounds. So if you only share the positive data, then you have skewed prediction, so a high sensitivity, but a low specificity. So there is a lack of negative data, not negative in the sense of Donald Trump, but in a true scientific sense. How can one turn Ames negative into a positive value for mutagenicity prediction? Quite simply by data sharing. And funny enough, this is a publication which you see on the slide where I am a co-author, and it's, even though from a scientific perspective, my contribution was very, very minor, it's one of my most quoted publications, simply because people use this data on and on for building Ames data prediction. Now, this subsequently entered into ICH M7. So one of the other co-authors, Andreas Sutter, can be found in this seminal guideline for testing or assessing DNA reactive compounds in the guideline of ICH M7, which came out 2014. So look at the time gap. 1988, Ashby and Tennant, and only in 2014, so almost 30 years later, we had the first guideline of in silico use, or for in silico use. That tells us that the whole sharing, data sharing issue takes a long time until you come up with meaningful systems and models. But this did not really frustrate us. Next slide. We're looking beyond data sharing, beyond mutagenicity, because we immediately realized that there is a big value in the pharmaceutical industry of all these non-published data, partly negative, partly positive data, not only for in vitro assays, but also for all the in vivo studies we do with the animals for submission. And the assets are, this is high quality because it's mostly done under GLP. You have the pharmacology included. It is guideline conform, so it is more standardized then compared to academic research. We have data on different species. We have a broad chemical space, multiple endpoints, and so on and so forth. But this is like a precious value sunk on the shores of South America, and you need to get it out because it's in paper archives, or at best in PDFs, but not really easily retrievable. And now, if you think about the time it takes to construct in silico models, then those who should finance such data extraction exercises, and next slide, please, would, where are we? Are we now in slide number 6? Or are you at 5? So please jump to slide number 6. I probably forgot to say next slide, please. Slide number 6. The value proposition, if you look beyond just in silico, because as we have seen, it will take decades until you come up with a validated in silico system, particularly if you now extend the objective to in vivo in silico prediction. But there is a clear value also in read across. Imagine the situation, toxicologist runs a new project and he remembers that he has seen such a toxicity already some previous years, or has talked about with a colleague about this toxicity. He wants to retrieve that. Now he could go to PubMed. He could also go into the archives and collect all the interesting publications, or look back into the original reports. That will take him hours at least, probably days, sometimes even weeks if it's a little bit more complicated, until he reaches a conclusion. So that's the clear value proposition, that if you have a repository with meaningful, decent search capabilities, then you will save a lot of time and consequently a lot of money. And this is actually the reason why, slide number 7, we started some 10 years ago this IMI project, ETOX, which stands for electronic toxicity, with 13 pharma companies, 11 academic institutions, to build up such a repository of the pharmaceutical industry for the legacy toxicity reports, which are not in the public domain. And from there, develop innovative in silico strategies and novel software tools. And the general objective is quite clear: to better predict toxicological profiles from small molecules in early stages of drug development. I'm saying early stages because it's not in the GLP environment, even though we collect GLP data. The systems per se are not GLP compliant. Next slide. What are the prerequisites for safe data sharing? You have several levels. There is a legal aspect to it, and the legal aspect has 2 components, one on the consortium level, and then on the level of the individual pharma company, where the protection of the intellectual property is key. Then you need physical controls. At the time of ETOX, the cloud was not really an option. In the meantime, this has dramatically changed. So at that time, we were talking about systems behind the firewall and a local service at the honest broker. And then you certainly need software control. So this is something we needed to develop at the start of ETOX. Next slide, please. And what we discussed internally with the participating pharma companies is that we needed several levels of protection, depending on the confidentiality level of the individual data set. So we have come up with 4 levels. That's the public data. That's trivial because you can collect it from various sources. You have just to have a look at the copyright. But then more complicated, if you go to the non-published company data, you have to decide individually whether this is shared non-confidential or whether it's shared confidential. Now, sharing confidential seems like a contradiction per se. But what we did is we put the same confidential data in the database of the honest broker, but only the company which provided that data could see it. Why did we do that? Because then we were able to align the ontologies, so the terminologies of the reports, with the confidential data. So we had the same search capabilities, but could then also see the bare data. Novartis could not see it. They could only see the bare data which we provided non-confidentially. And we could later on decide, for example, if we said, okay, it was in the past confidential data, but now the compound is dead or it's on the market for a long time, we switch it to the level of non-confidential and share it with the whole consortium. So this is the reason why we put it to the honest broker, even though it was confidential. And then there was a fourth level, which was exclusive private data. We collected it internally, but did not send it to the honest broker, tried to create it to the same level as the honest broker did, and then use it for internal model validation, but the models would not see it. These categories were implemented in the consortium agreement. So all participants from the FPR side agreed to that terminologies, the lawyers internally signed it. So that was the general framework. Next slide, please. But that does not really tell you what the company internal policy is with regard to IP protection. So what does the company decide what is confidential and what is non-confidential? The policies are not streamlined across the different participating companies, but more or less many companies followed what we did at Bayer. After long discussion, what is actually the IP value in the toxicity report, we concluded it's essentially the chemical information. So the tox finding per se does not really carry an IP value if it's not combined with structural information of the chemical or the pharmacology. So therefore, we had this tiered approach for releasing a compound into, or a tox report into the non-confidential area, where we said, is there a CAS number, chemical abstract service, is there a CAS number already existing for the compound? Because that would only be the case if the compound is patented and sufficiently advanced in the development so that it would already be protected against competitors. If that was not the case, the compound was not eligible for the non-confidential bucket. Secondly, we had to discuss with the head of medicinal chemistry whether the compound is of a particular interest. That is a vague criterion, but an example for that is if a compound is under litigation, we will most probably not share it, even though it had a CAS number. After that check, we had to check whether there is an IP conflict due to collaboration. So if he had sold the compound or partnered it out, we needed to check it with the partner, whether he agrees to the confidentiality status and would be willing to share it in the consortium. If that was also positively answered, then the status was non-confidential, and if there was any no, the data remained confidential. And as I said, the small arrow in the bottom tells you that this decision from confidential to non-confidential could always be revised, logically not the other way around. So if it was once non-confidential, then it was more or less gone in the consortium. Now, to make a big jump of what did we achieve with all that data sharing, we built up in eTox a repository with more than 1,800 compounds and more than 8,000 toxicological reports, and developed what we called, and this is seen in this slide, eToxis, which is slide number 11. It's a one-stop shop for both the database query, but also the prediction systems we built in the course of the project. And to give you an idea how the system actually looked like, that's on the next slide, slide number 12. Essentially, you could query the system, you can query the system with structural searches, which includes also moiety search or similarity search. Then in the middle, probably a little bit hard to read, you could search for specific pharmacology. And then on the right hand, you could query for the toxicology in a rather sophisticated way. So you could drill it down to specific liver findings in the histopathology for the specific species. And you could certainly combine the individual query fields. So structure plus species, structure plus pharmacology, but not in a certain species, and so on and so forth. And an overview publication is on the screenshot on the left, right hand on the slide. And if you go to slide number 13, that gives you a flavor of the different predictive systems we have developed in ETOX. So, for example, highlighted here are systems, predictive systems for absorption. And again, you type in or draw a structure. You could incorporate it from with a SMILES code or other codes, and then run the query and get the results. And on the right-hand side, you see the model documentation, who has prepared it, what the prediction values are, and so a basic level of documentation. Now, what would you do with such a system? And a case example is shown on slide number 14. To illustrate, particularly in the early phases where you are in strong collaboration with the medchem people, how you could use the system. And this is a true case example where a medchem person came up to me and asked me, is there any difference between metafluorophanyl substructure, which I attached to the pharmacological core, or parafluorophanyl with regard to liver toxicity, because there was a suspicion from other studies that there might be an issue with liver toxicity due to metabolism. Now, if you go into in silico systems for DILI, you do not really get a clue, because it's not very well covered and the mechanism is not so well described. What we did was a purely statistical approach. We looked for compounds in the database which had this moiety, the two moieties, and looked for several indications of liver toxicity. So, for example, liver histopathology and transaminase elevation, which is shown at the bottom on the x-axis of the graphics. And we matched it for the compounds and looked then how often does that occur overall in all the compounds in the database. And I said 1,800 compounds, but you see a slightly higher number, because here also the proprietary compounds of Bayer are included. That's why the N is 2,544. And if you compare the two graphs, you realize that the metafluorophanyl has higher yellow bars compared to the blue bars. So the incidence is relatively higher compared to the background, so overall occurrence in the database, which tells you that evidently the metafluorophanyl tends to have a problem with liver toxicity. And I deliberately say tends, because it's not really proven, but it's also interesting that this moiety is with N equal to 13 underrepresented compared to the parafluorophanyl, which has 94 hits in the database, but in terms of incidence of liver signs, is equal to the background. So this is a use case how you could, from a statistical or read-across perspective, use the database to make first assumption with regard to organ toxicity. And this has been published in a publication which I did together with my colleague and leader of ETOX and also eTranslate, François Pognon. But if you click once more, Connor, but what is the ETOX animal data? Is it really predictive for man? Because at the end of the day, it's nice to see a prediction for animals and you get a certain clue and you have the suspicion that it correlates with men. But particularly for liver toxicity, we are aware that the animals are sometimes over-predictive, but also very often under-predictive. So the question is, what is the concordance of our data with the clinical data, which we did not collect at the time of ETOX? I had a discussion with colleagues from Elsevier, which provide Pharmapendium, which is illustrated in the next slide, 15. Pharmapendium collects both the clinical adverse events and the preclinical adverse events. And Matthew Clark, which is a brilliant Pharmapendium expert, he should be because he's at Elsevier, but he has also a tremendous statistical background. So we said, let's mine Pharmapendium and redo a little bit what Olsen did in 2000, the seminal publication about concordance preclinical data, clinical data, where Olsen at that time used 130 compounds and matched it, really paper-laying report, clinical report, next to the preclinical report and said, is it concordant or not? We did it on a statistical basis, using more than 1.5 million adverse events reports, and you see the count of observation in the right table, and did this statistical analysis, which was published 2018. Just to give you an idea what you could do if you collect all the data and do not have to look across paper report by paper report. And the outcome from a scientific perspective was not so surprising. It's just the methodology. So we confirmed more or less that animals do predict human toxicity with certain limitations, and many of the limitations have already cursory, on a cursory basis, been reported. If you go on the next slide, that's the way we depicted our findings. So if you look at the picture, you first look at it from the human side. You see the little icons from the animals, and then the individual findings. It's extremely hard to read. So if you want to read more, you have an animation limited to the monkey. And here, this picture illustrates how well the finding, for example, somnolence in humans is predicted by the monkey. And that is almost trivial finding. Somnolence is best predicted by monkey, not so well in rats. Everybody who has a decent understanding, and you do not need to be a physician or a toxicologist, would have predicted that. But this is now done on a statistical basis. So you could go back to Pharmapendium and retrieve the individual reports which contribute to this statistical correlation. Another less known phenomenon is skin disorders, which are very well predicted with monkey, not so well with rat. And this is for those who are in oncology research, particularly in EGF inhibitors, are aware of that, but this is now also proven by this broad statistical analysis, which shows us that such a big data approach is extremely valuable, and which actually also drove the extension of the eTox project into the follow-up project eTransafe, which was started in 2017. And eTransafe clearly means that we now are looking for the translational aspect of this data collection. We want to connect the preclinical data with the clinical data. This is now slide number 17. And if you go to slide number 18, this gives you an overview of the concept of eTransafe. So we want to make the preclinical safety data fair, which at the time of eTox, the concept didn't really exist, verification of data. And we want to correlate with clinical data, so a big data translation analysis. And on the right hand, the sketch illustrates a little bit what you should be able to do with such a system, which we call Knowledge Hub. On the one hand, you have the typical query system similar to eTox, where you start with a compound, where the main customers would be the medchem people or the toxicologist. But then you could also come from the event side, so where you observe in a clinical trial or also in pharmacovigilance, a certain human event, and then you go also back into the database and search whether there was any indication in the preclinical species and any indication in the same field of indication, the same target class, or so on and so forth. So that's the objective. And certainly, we also want to model the findings, so build predictive tools as we did in eTox, but now also including the clinical part of the data. Next slide, please. Slide 19. This gives you, for those who are the IT aficionados, a little bit the overview of the IT architecture. And if you have any questions to that slide, I would probably refrain from asking them, because that's something I'm not really familiar. Just like the slide because it's so fancy, so modern with all the icons. But essentially, now it's a cloud. It's a cloud architecture, no longer behind the firewall. you need a secure authentication and access from outside. That's what we have discussed in one of the first slides with regard to IT protection levels and what you need in the architecture. We have deployed so-called primitive adapters. Primitive adapters, that's something I learned in the course of the project, is the protocol for the interfaces when you want to hook up to other databases which are outside of your project, like, for example, the FAERS, FDA FAERS pharmacovigilance database, or clinical trials databases, and so on. There is Flame Service. Flame Service is a development in eTransafe where you could, for example, do a similarity search, but you could also train your models with data which you do not share in the project. So the data you use for training or retraining the model is hatched. Nobody can see it, but the model is then feedback to the whole consortium. The setup of shared SQL and web I use, this is, as I said, a little bit strange vocabulary to me. There are data browsers, there are can search and quick search. The can search, in order to speed up a can search, is a little bit what stands behind Google. If I dare to say them, somebody would probably object to that simplified statement. Next slide, please. For sharing the clinical data, we were thinking of applying a similar tiered approach, and we were starting from the preclinical data. And this is again the Bayer's perspective, but most of the companies have done similar approaches for data sharing. And the confidentiality level can already be triggered by the question: have you already shared preclinical data? Because then the structure is already out. The structure in clinical trials, in most cases, in almost all cases, except if the patent lawyers have committed any fraud, is already protected. So this should not be a problem. However, then another problem arises, GDPR, Global Data Protection Regulation, because you have sometimes individual patient data, and this needs to be covered by the informed consent. However, we are collecting eTransafe aggregated or anonymized data. So if that procedure has occurred, if the data of a clinical trial has been aggregated and anonymized, we can share it. Then again, IP check, because in the meantime, we might have partnered out the project. So if that is the case, we need to ask the partner or leave it in the confidential bucket. Then the question is, the clinical data already in the public domain, such as OIDRUG-CD or clinicaltrial.gov, then it's clear that we can share it. And this procedure there is a statement of two names, which is only relevant here internally. These are the bosses who decide about this procedure, and therefore they agreed to that, so that I'm on the safe side whenever I run the procedure and share the data. So forget the SOP, that's Bayer internal, just take as home message a procedure which has been cleared by Bayer internally. Next slide, please, slide number 21. This shows how important all these internal procedures, this legal framework, the IT architecture is for speeding up a project. If you look in the time course of data collection, so on the x-axis you have the year of the project, and the eTox project initially ran for 5 years and then got a 2 years extension, so overall 7 years. And if you look in the data collection, the count of the nation of studies, then you realize that it started only in year 2.5. So at that time, half of the time of the project, which made us extremely nervous, because we said, how could the modelers start to model if they don't have the data? But the issue was that all the debates with the company, internal patent lawyers, IP protectionists, the project managers and so on, but also setting up the legal framework and the consortium took us more than a year. And only after 2.5 years, the project took off. And if you click, there is, I think, no, there is no animation. If you compare it then with the onset of EAT-and-SAFE, there was already substantial data contribution after 5 months, half a year. And the simple reason is because we took over all the legal framework, the internal IP clearance procedures from ETOX, and then had more or less a head start in ETOX. And now, after almost 2 years, have achieved a similar number of donations we had achieved in ETOX year number 7. Even though now we are collecting individual animal data, which we didn't do at ETOX, we are collecting send files directly from the LIMS system. So these data donations are much more granular compared to what we have in ETOX. So higher granularity, higher speed, and at the end of the project, also much more data donations. Next slide. Now I have an animation. So this previous experience and implementation of best practice policies improve the speed of data sharing. Question to Connor and Barry. I do not know how much time we have left. We could make a stop here and open the webinar for questions. I could also continue with a couple of more slides.
No, Thomas, I think go on and finish. It's fine. It's good.
Barry, I'm not sure whether I understood you correctly. Shall we finish here and ask questions?
Yeah,
or
let's go on and finish. You've just got a couple more slides to go. Let's do that.
Okay. Now, we had a couple of examples what we can do with the shared data from ETOX, but I want to continue with what we have achieved and what we do with the data in EAT-and-SAFE. And we have presented the midterm review of 5 fields of innovation. I will not talk about advanced in silico model. I'm now at slide number 22, and if you click once more, the in silico models will disappear. I will mainly talk about the study report domain. I will talk about the Rosetta Stone and virtual control group and good practice in data sharing. The last point is a logical extension to what I have already said in my talk to develop guidelines. Let's start with new standards and new tools. The general issue in setting up a database, now also with the SEND data for toxicology reports, is that you collect the SEND data, but you do not really collect the basic conclusions out of the report. So particularly you miss, if you only use the SEND data, you miss the conclusions of a study director, what he concluded was treatment related and not treatment related. And this is an extremely important information. We already had discussions with FDA and CITES. Why didn't you include that domain, study report domain with treatment related? And they said, because at the beginning we didn't think about it. We have started to develop that. And if you click now through slide number 23 with the 2 animations, what we now do, we developed a text mining pipeline for treatment related. So we have recently built a corpus out of 90 reports where toxicologist experts highlighted, what you can see in the middle, highlighted the doses and treatment related and the individual finding. And then a so-called study report domain, CITES-like, in the same format was developed, which can later on be automatically be incorporated in the database. So you capture this information and you keep it. Yeah. And in the meantime, we have also developed a tool which can be used while report writing. So while you type your report, you could also fill the study report domain and connect it to your SEND data. Even though FDA is not yet requiring it, you can fill your own repository or the EAT-and-SAFE repository with that study report domain. So you can only do that with shared data or shared reports, because then you can build a decent text mining corpus with the relevant tools. If you do it only with your in-house report, you don't get very far. The Rosetta Stone, when you talk about, and this is now slide number 24, when you talk about concordance analysis, and I already mentioned the seminal Olsen study 2000, where he manually curated and assessed 130 compounds for the preclinical to clinical concordance. You have a lot of manual curation effort, and you got also stuck in the different terminology used by physicians and toxicologists. So big data analysis can only be performed when you have common terminologies, vocabularies, and ontologies which align or link the different ontologies. And this is a little bit what I have already talked about, this publication we did together with Matt Clark. We call it the Rosetta Stone. You're probably familiar with this excavation done by Napoleon soldiers in Egypt, where they found the stone which had, on the one hand, if you click through this animation on slide number 24, you see a picture of the Rosetta Stone, which has the hieroglyphs, a demotion language. I didn't know what that is, but I had to look it up in Wikipedia, and the ancient Greek. And the ancient Greek was essentially the key for deciphering the hieroglyphs. And this is a little bit the metaphor we are using now also for this concordance analysis, because we are now lining up the clinical data, which is most often in MedDRA. I was thinking about the hieroglyphs being sent, because if you see the send terminology, it essentially means nothing to you. You have to link it to another terminology, and this could be SNOMED. And that's essentially the translation to jump back and forth between the different terminologies and the different assays, preclinical and clinical-wise. If you go into slide number 25, that's the illustration what we want to do. In the middle, you have this alignment, and if you come from the right, which is the preclinical terminology send, you can link it up and drill it down. For example, you don't have histopathology. Most often you don't have histopathology in clinical trials, but you could link a histopathology finding in an animal study to a certain terminology and then link it to liver findings in the clinical study. So that's the whole idea. Deciphering the Rosetta Stone will enhance the translation analysis. Yeah. So that's one aspect for the use of shared data. Another one is now slide number 26, data sharing and virtual controls. We were recently discussing in eTransafe, could we also share data from reports where we don't share the pharmacological or chemical data? And why would you do that? What would you do with the data? There is all this animal control data, which is highly relevant, because already in clinical trials, people are discussing we could use virtual patients, virtual participants, so constructed simple by electronic health records. Similar approach could be used for preclinical trials, so for animal studies. The historical data in the companies is anyway collected, because you already use it in order to assess whether a certain effect in a study is in the range of the historical control, or is it really outside. And in compared to patient, animal studies are much more standardized and controlled. So you can easily use this data and potentially replace 25% of the animals, that's the size of control group in the regulatory study, with electronic data. So replacing control groups could contribute to a reduction of animal use. And when we discussed that internally, and also then with a couple of CR experts, there was great excitement. And this is illustrated in the next slide. It's not an easy task. If you go to slide number 27, which illustrates a little bit the structure of a 4-week toxicity, the number of animals, in this case, non-rodent rats, 12 in each group per gender. And if you now take the control data, if you click corner once more, if you take the control data and put it into a historical control data collection, share it, and then do a thorough statistical control where you determine the thresholds, you would allow for data variability. and then use it in a randomized way to collect a synthetic control group, you could eventually replace this control group. And this is the reason why I say 25% of the animals could eventually be replaced and saved, because then the control group is entirely electronic. As I said, the idea has received broad repercussion in the 3R community. As a consequence, if you click once more, we have published the concept in alt text, and it was actually Thomas Hartung who asked me, please make a food for thought article out of it, because it's really an interesting idea. Yeah. And this is now the penultimate slide, which concludes a little bit the session. We will continue to write down our experience and cast guidelines or white paper out of it. One has been drafted. It has a little bit been delayed because one of the main authors was involved due to COVID-19 in other activities. She was not ill, but the activities of Elsevier, of Elixir, in this case, Elixir, she's working has been shifted a little bit. So we originally wanted to submit it in May. Now it will probably be submitted in August, end of August. But essentially, it's a first step to line out a potential future guideline of data sharing, which then comprises the main factors I have told you in this talk. Last slide, in kind of a summary. Data sharing requires a legal framework and technical solution. Data requires verification and sometimes a tremendous effort of curation. Data sharing also requires changes in mindset, and this is actually not trivial. You can set up the best legal framework, but then you have internal experts who think this is my data. I did the experiments. I'm the only expert who can interpret it, and believe me, it is a serious problem, not only at Bayer, in each and every company. And this is something you can only, you have to massage these people, but you also need really bosses who are into digital transformation and tell the people, this is not your data, this is company data, and please let it go. Yeah. Your data are assets. What is the return of investment for sharing or data sharing is worthwhile? You need to share the use cases and success stories, particularly in order to convince those people I just described. And on the right-hand side, you see a slide I stole from Martin Roche, who is a data expert there, and he compares the whole thing a little bit to gas exploit or oil exploitation, where all this valuable fuel is burned because there is no technical solution for using it. And he uses this metaphor of burning these gas in the refineries and puts data in each flame. And he also provided a figure for these 147 billion of square meter of natural gas, which are flared in 2015. And asked the wonderful question, how many data assets vanish every year because we have poor data management and cannot really share it, neither internally nor with partners. And with that, I'm done and happy to take any questions.